All right, so this short video lecture is meant to wrap up some of the review topics that are from prerequisite courses to our Fundamentals of Biochem class. So this will be very short, again, just some key points that if you aren't familiar with, please refamiliarize yourself with or contact me to discuss further, right? So these will be topics that we see on our first homework assignments, and that are, essentially a foundational part of things we'll talk about further into the semester. So just our general overview, right? If you haven't watched the central dogma of biology, air quotes around dogma, if you haven't watched that video or link on our Brightspace page, please, please take the 5 to 10 minutes to do that. There are some very key steps about, essentially, again, every step along the way that we're going to be talking about in class, as well as some key tools you might be using in lab. It's imperative that you have a foundational and basic understanding of the topics in that video. So go ahead and check that out. Again, it's 5 or 10 minutes. It'll help you greatly for lecture and help you greatly for lab. And in that, that's going to reemphasize why we care about DNA and amino acid sequences. There's some, again, review and emphasis in that video. Please check it out. So what we're going to talk about, again, very briefly today, just some concept about protein and protein function, how proteins behave in a cell that you may not have thought about before. A very brief intro to signal transduction if you've never seen it before. Again, we'll go into more depth about this later in the semester. This is, again, just a catch-up. You should have these basic understandings when you enter this class. And then a loose discussion about cellular structure, right, where we can expect to see some things in cells. So one term that we'll see come up a couple times throughout the semester when we talk about protein function as well as diffusion through a cell is macromolecular crowding, right? So macromolecular crowding is essentially a phenomenon where you have a low overall concentration of something, right? So, low concentration of something. There aren't very many of them in a large volume of say, solvent or, say, liquid within inside a cell. But even though there are a small number of them, they're very big, right? And so in this case, within a cell, we're typically discussing proteins, right? Compared to the total number of salt ions, whether that be sodium, potassium, calcium, magnesium, etc., the concentration of proteins is usually lower, but they are very, very, very much bigger compared to those small ions, right? So over here on the top right of our slides, we have this three-dimensional image, I guess if you want to call it, that's meant to represent, what the density of proteins and the structure of those proteins within a cell might actually look like, right? So all these small globular things here are meant to represent various proteins. And if you look closely, you can see that there really isn't that much empty space, right, in between these proteins. So it's not as if the inside of a cell is just a void of water or solvent. And so down here in the bottom right, we also have these cartoons that are meant to represent how diffusion within a cell might actually work, right? So in here on these two images to the left of this figure in the bottom right, you can see that this piece of solute here, if it was to find its way into a cell that was completely devoid of proteins and things, you may expect its diffusion pattern to be, at random, but eventually it could cross the entire cell. The same could be true about a signaling molecule, right, or a biochemical reaction. If there isn't a whole lot in the cell to take up all that space, whatever signaling molecule you might be talking about, or ligand, or something else, is free to diffuse throughout the cell wherever it sees fit. However, if you clog up the inside of that cell with various proteins, in these two images over here to the right, you can see that the diffusion pattern you might actually see for a solute, or again a signaling molecule or a ligand, is actually quite restricted, right? Those molecules or solutes aren't really free to diffuse freely throughout the cell. And then over here in this small GIF, right? So this is actually a glass of warm water with some green food dye, and then a glass of cold water with some green food dye. And I just have these in here as a visual representation of maybe what you would expect to see in a cell devoid of proteins, right? You would expect to see much faster diffusion, as if, similar to what you'd see here in the hot water, versus a cell that's full of proteins, you would see limited and much slower diffusion, right? So hopefully these visuals might help you. And before we get into proteins, right, in our third or fourth section, just some definitions, right? So living cells have unique sets of proteins. It depends on the type of cell, right? You wouldn't expect to see the same overall number or types of proteins in, say, a cardiomyocyte as you would a hepatocyte, right? So basically a muscle cell in your heart versus a liver cell. You wouldn't expect those to express the exact same proteins. And beyond that, the types, numbers, and everything else about the proteins are constantly undergoing change, right? So the proteins, much like the larger organism, is always undergoing some sort of homeostasis, right? And when we refer to proteins, we actually call this the proteostasis, which is just an elegant word for essentially the network that controls this proteostasis network. So again, just some review here, hopefully that you've seen in either cell and molecular biology or some of your other biology classes, right? So I'm sure everyone's heard of metabolism, but perhaps what you haven't heard of is how metabolism actually breaks down, right? So metabolism is actually a whole-sum game. It's actually a composition or a composite of anabolism, which is typically pathways that synthesize new things, right? So building things up, versus catabolism, which is actually usually degradation pathways, right? So you can think of anabolism or anabolic pathways as building things bigger, and catabolism is usually breaking things down, right? So this will come up a lot more once we get into the second portion of our class, where we discuss metabolic pathways, where we're either building biomolecules or breaking biomolecules down for energy. And what goes hand in hand with metabolism is also signal transduction, right? So we'll have a more in-depth sequence about this later in the semester, but hopefully you've seen this topic before, right? So signal transduction is essentially an oversimplification of how information outside the cell triggers and activates changes within the cell, right? So cells have an inherent need to adapt to many, many, many different stimuli, whether that's stimuli that perturb homeostasis or signals that actually help regulate homeostasis, right? So there's some basic concepts for signal transduction, a few regular phases, right? Reception phase, where a ligand or a signaling molecule or even a drug will bind an extracellular receptor or even potentially an intracellular receptor, and then a transduction phase, where the cascade, there's essentially a downstream cascade of intracellular responses. So over here in this generic signaling pathway on the right, we have our ligand or our signaling molecule that's going to activate an extracellular receptor that's going to activate some JAK-STAT signaling cascades downstream, where ultimately there's an effect within the nucleus of a cell and something about gene transcription changes, right? Again, a generic example. When we cover this in class, we'll cover it in a little bit more detail. So some of the basic phases that you can further break those two broad phases into are the four or five here, right? So four here. So reception is the first phase, right? So reception is when that signaling molecule or drug or ligand binds a receptor. Again, that can be an extracellular receptor or an intracellular receptor. Transduction, which is the second phase, where that ligand binds a receptor. That receptor undergoes some sort of three-dimensional change, which activates intracellular pathways. And again, this is so diverse, it's hardly worth saying much more about, but the activation of perhaps that single receptor leads to the activation of many, many, many second messengers, right? And that's the transduction phase, where one activation of one thing leads to the activation of many, many more, right? And then the third component of this is response. So response is essentially whatever the goal of activating that receptor was, right? Whether that's changing gene transcription, maybe even turning gene transcription off, activating a single protein or activating multiple proteins. Again, response is a very broadly defined thing that occurs after that extracellular receptor binds the signaling molecule. And then lastly, termination of the signaling cascade, right? So typically we don't want activation of these receptors to result in permanent activation of anything, right? We don't typically want a permanent response. For example, if an extracellular signaling molecule bound a receptor and the response of that was to cause a cell to undergo mitosis, you wouldn't necessarily want mitosis to occur indefinitely, right? At some point, we want to turn off or terminate that signal and stop our cell or cells from undergoing mitosis. Otherwise, you can have negative impacts that ultimately lead to certain types of disease, such as, in this case, potentially cancer. So up here, again, this is just a very simple 1, 2, 3, 4 cartoon or illustration of signaling cascade. And then down here in the bottom right, we have some examples of much more broad signaling transduction pathways. Again, we'll cover this in more depth when we get to our sections later in the semester. We'll talk about tyrosine receptor kinases, as well as GPCRs, hormone receptors, and a few other things. Right? Okay, so we've talked about a few of those, right? Some more, review for vocabulary, right? Hopefully everyone remembers their protein pathway feedback controls. So the first one that we'll talk about and give an example for is negative feedback, right? So this is when the products from a reaction cascade or a signaling transduction cascade turn off that same cascade, right? And then contrary to a negative feedback, there's positive feedback, right? So where products from a reaction actually increase the reaction cascade. So up here I have this domino GIF, right? So you can think of these as the two different aspects or two different parts of this domino cascade, right? So initially activating or knocking over that first domino sets off a positive feedback loop where one domino triggers into the next, and you have this large giant cascade of more and more dominoes falling. And then ultimately, once the last domino falls here, right, in some ways you could think of that as negative feedback, that the end of the process has ended the whole signaling cascade. Right, and that's probably going to keep going here in the background. But if the domino visual doesn't really help you with that, we have some cartoons down here, right? So on the left here, we have, say, enzyme or protein A, right? So we're going to pretend activation of enzyme or protein A activates enzyme or protein B. Enzyme 1 is going to react with reactant A, which will produce reactant B. Reactant B will interact with enzyme 2, et cetera, et cetera. And what you see is the production of many, many, many products down here, D, right? And as the concentration of product D increases, that's going to inhibit enzyme 1, which if we scoot over here to the right, after we inhibit enzyme 1, we get basically a slowdown of this pathway and a limited production of D. So this would be an example of negative feedback, right? Versus over here on the right, we have the same pathway where with A ultimately leads to the production of D. However, here, an increase in the production of D actually activates or increases the activity of enzyme 1, which, again, scoot to the right, will ultimately lead to the increase, or you could think of cranking up this pathway, will result in the increase in the production of D. So here, positive feedback, producing the product D results in producing even more product D. Right, and we'll talk about a few more of these specific examples when we get to cell and signaling in Chapter 8. So that's it for this very short video. If you have any questions, shoot me an email. Otherwise, I will see you in class.